Everyone sees that? We good?
Yes,
Okay.
I can.
All right. All right. Well, thank you, Tristan. Thank you, Serena, for putting this together. And even though it's early in California, I'm really excited to share with you all say some of my thoughts on the CA2. For the interest of time, I'm just going to talk about, and the special issue I talked about gene expression and connectivity. Today I'm just going to talk about some of the gene expression in CA2, and Serena's already kind of touched on. Okay. So my interest in CA2 really started when I was doing the study on Hippocampus and trying to create an atlas, a gene expression atlas of the whole hippocampus using the Allen Institute brain data. And so my approach to creating this atlas is kind of described here. And this is just a little example, but what I call gene expression pattern mapping. So at this kind of rostral level of the hippocampus, this is basically 15 different gene expression in situ hybridization experiments from 15 different brains. So there's the top row you'll see here are genes which are expressed kind of in all of the pyramidal cell layer in this area of the hippocampus. The middle row you'll see is only expressed in kind of the top half of that pyramidal layer. And the bottom row are only expressed in the bottom part of that pyramidal layer, right? So the idea here is you can see that there's essentially a boundary, right, between the middle row and the bottom row that creates this line I've drawn through here. That a boundary is kind of reflected on both sides, right? So that there's complementary patterns, and really what this also means is that some gene expression in the hippocampus is combinatorial, right? So the top row can be understood as being a combination of both areas, whereas the other two are more distinct. So taking this approach, I then went through the entire hippocampus for all of the mouse rostral caudal levels and coronal sections to create this atlas and define 20 distinct kind of molecular domains through dentate gyrus, CA3, CA2, CA1, and the spiculum. So this allowed us to kind of put together, the 3D model of the mouse hippocampus. And what I want to point out here is CA2 is that thin orange area that you see in the front. And even though in the kernel sections, it often looks kind of small, it's very obvious when you look at the 3D model how long it really is, how far it extends. So this idea of the CA2 really kind of expanding beyond its borders, really, as Serena mentioned, started with Edwin and Allen Institute first noted this. And I kind of want to just go into some more of the details of what I saw in the gene expression of CA2, how I drew the boundaries in the atlas, what are kind of the key features I can see. So I kind of treat this level is kind of, to me, what I call the classic level of the CA2, the one that we most recognize, where you often have in mouse this kind of bump in the pyramidal layer. And when you look at some of the gene expression markers for this, as Strange showed, there is some variability kind of where in this bump goes. To me, I often see it mostly right starting kind of at the 2/3 proximal bump and then extending down towards the CA3. And the reason to think that these are good gene expression markers is that it's spatially located where Lorente Denel first described this based on the Golgi morphology. And I think most people now have seen that there are plenty of gene expression markers for the CA2. Actually, to me, is looking across the hippocampus, there's a lot of really good markers for CA2, and that might even be something unique for CA2. But we can look at all of these markers and there are differences between them, but there are also a lot of similarities, right? We can consistently see kind of the same pattern being reflected by all of this and have a kind of consensus gene expression pattern, right? And so in all of these, this is kind of the example of the classic level, but in all of these, you see that the gene expression extends forward rostrally as well as caudally from these levels. Now, some of the variations you can see here can come down to just the differences in the oblique sectioning across the brains. when you're comparing across lots of brains, sectioning can kind of shift these boundaries because rostrocaudally, the borders of the CA2 will shift dorsal ventrally as you go across rostrocaudal axis. So that's something to take into consideration. But there's also variability, right, in this gene expression density you see, right? Some of these markers are more dense, so they're stained more densely, they're more highly expressed in these cells, whereas others are less dense, right? So it suggests that there's a lot of cellular heterogeneity here. And I think what Serena started to touch on is if you start overlapping or co-staining a lot of these markers, do they actually show up in the same cells, or are there actual subtypes, right? Subtypes of cells which co-localize certain markers, others which might be different, right? And so we want to know is there a gene expression profile that defines CA2, maybe not based on one marker, but several markers. So I just want to kind of touch on essentially first, where are the boundaries of CA2 in the atlas? And I just wanted to show, so this is kind of now moving the new CA2, starting from the kind of more the classic level in the bottom section here. You see the gene expression on the right. This is for Amigo2, and I just picked one of the markers to use here. I'll kind of mix and match several markers just to kind of make sure you guys can see different levels. But the thing you see is that the CA2 labeling, first as you go rostral, you see both kind of this pattern on the medial and lateral parts of the area, right? And you see it more at the classic level. You see the fasciole cinerum has a lot of the CA2 gene expression. That's very common. A lot of the markers for CA2 also are expressed in the FC medially. And as you go rostral, that FC starts to disappear, and this gene expression pattern kind of flattens to become more what I think is part of CA2. And as you go even more rostral, the CA2 kind of starts to close in on the CA1, starts to come across. And so that corresponds in my atlas to the orange areas, right? The orange area here on the left is the CA2. Now, if you compare that to the Allen reference atlas, that's pretty consistent. That hasn't really changed too much. But what I want to also compare is the newer Allen CCF version 3. This is the Common Coordinated Framework, which is a 3D mouse atlas that's being used for Lightsheet. And there are a lot of differences with this new atlas in the hippocampal boundaries, the delineations, and particularly for CA2. And so you can see CA2 here, as I've shown it, is really much thinner. So it's important to kind of be aware of these differences, as you're using different atlases for analysis. So going even further rostrally, the CA2 gene expression kind of closes in on the top, and that's where you get this kind of continuous gene expression pattern across. Ultimately, the CA3 kind of comes up and closes around it. And then if you see the very rostral tip, sometimes you'll find some labeled cells there. It's mostly CA3 cells. So I've kind of, in my atlas, I have the CA2 end the section before that. But you could also make the argument that CA2 maybe gets all the way to the end. Comparing that to the Allen reference atlas, you see that doesn't really match. So the Allen reference atlas, this is version 2, doesn't have the CA2 continuing here. They have this mostly as CA3. The CCF version 3 does have CA2 here, but again, it doesn't really match the pattern of labeling that we just saw in the gene expression. It's I think, in fact, identifying more of the stratum lacunosum moleculare level rather than the true CA2 boundaries. If we look, that's the rostral extension. If we look more caudal to the classic level, you have to see the CA2 boundaries shift ventrally, and ultimately come to this level here, which is level 81 in the atlas. And here's where you see the CA2 really kind of dive down ventrally. And the question for me was, how far ventral does the CA2 go? You can see the PCP4 here. How far does this boundary go? Does it go all the way to the ventral tip? I don't think it does. I think that the ventral boundary of CA2 is somewhere around kind of adjacent to where the endopiriform, the dorsal endopiriform nucleus is, somewhere around this level here. It's sometimes difficult in with the sectioning, especially with oblique sectioning in the dorsal ventral direction for when you're cutting coronally, how that can change. So I put it there. It's interesting because it means that the CA2 is almost always interposed between CA3 and CA1, except for the ventral parts of the CA3 and CA1. So, but almost, one section is 100 microns later, the CA2 almost completely disappears. And then, except for a few cells, you'll see in the deep part of CA1, you can still see that. And that's something I saw both in coronal and sagittal, that there are deep cells, kind of CA2 labeled cells, the deep part of the intermediate CA1, and then 100 microns later, they're gone. So again, if you compare this to the Allen Reference Atlas, they don't fully, it has the CA2 there in these levels ending, so the kind of the caudal boundary is consistent, but they don't really show the full extension of the CA2 in its full depth. I have it going much deeper. And in the CCF version 3, again, the CA2 they have it going much further caudal, but again, not really lining up with what CA2, the pyramidal layer is, and it's more, I think, identifying the stratum lacunosum moleculare. So just important kind of comparisons of, I think a lot of people think, oh, well, we already know the boundaries of CA2, but really there's differences between atlases and what's used, and I want people to kind of be aware of these differences when they analyze their data. Okay, so moving beyond that, the question I wanted to get into, I kind of played with in the commentary was, are there different cell types or CA2 subtypes, and do they exist differentially along these borders, right? If it's extended, is there differences kind of that we find? And there is evidence already, I think, from Lissette and Stephen Siegelbaum's lab for functional heterogeneity of cell types. So what I wanted to look at, so I kind of took these gene expression markers to a single cell sequencing data set. So I used the DropSeq data that you can find on dropviz.org. This is from Steve McCarroll's lab. There's a very useful website there where you can kind of investigate their DropSeq data. And so what they've done is, they did the whole brain and they cluster it, and they identified this major cluster of CA2 and CA3 glutamatergic neurons. And then they cluster again, and they find 8 subclusters. So these will be 8 putative glutamatergic neuron cell types. The names here are essentially named by what is the highest expressed differential protein, but I want to make sure that RGS14 cell type does not mean RGS14 expressing. It's not, the other cell types also express RGS14 in variations, but we'll see that in a second. So the naming convention can be a little confusing, but these are, for the sakes of the argument here, the subclusters are essentially cells or groups of cells. So what I want to do is investigate, well, what do our marker genes look like expressed across these 8 cell types? I assume that some of these clusters, there's going to be some CA3 and some CA2, but we've already kind of said that these markers are somewhat CA2 specific. So we can kind of hopefully try to weed out the CA3 cells and look at the CA2. And so the thing you see here is that these are 6 kind of marker genes identified: RGS14, Amigo2, PCP4, MAP3K15, CACNG5, TM2. And you can see all of them, there's differential expression across these kind of 8 clusters. The PCP4 and Amigo2 are kind of the most broadly expressed across 8 clusters. It makes sense. Amigo2, I think, also has some ventral CA3 cell type staining in there, so that could also be included. But you see that those 2 are maybe the most broadly expressed across those 8 clusters. RGS14, like I said, the first cell type here gets named that because it's the highest differential expression. So that 3.3 value versus kind of the other expressed values is somewhat low. The difference between that's something like 2. So you see it's pretty strong, but it's also not exclusive. So the interesting thing for me was the other genes here, MAP3K15, CAGN5, TM2, which are lower expressed than some of the other ones, but also a little bit more specific. So MAP3K15 is expressed in this RGS14 cell type, weakly expressed in some of these others. CACNG5 and TM2 really only expressed in that one cell type and not in any of the others. So there's weaker expression, but somewhat more limited to a specific cell type.
Mike, can you wrap it up at this
Yeah,
point?
yeah. So this is we're going to end here. So I just wanted to look back at the tissue and see, you know, are these 8 cell types then distributed differentially along the CA2 axis? And so kind of looking now at the sagittal tissue sections that I showed here, there's a kind of, you can more clearly see the dorsal and ventral CA2. And, you know, for these other gene expression markers in the bottom, you can see that there's expression in both the dorsal and ventral parts. But for these 3 gene expression markers, you see a similar pattern of expression where TM2 and CACNG5 aren't really expressed in the ventral, right? So you see something similar to what you're seeing here with the RGS14s only expressed and then nothing in the other cells. MAP3K15 has this lighter expression, and you do see, if you look in real close, there is some very weak ventral CA2 expression. So kind of putting these 2 things together, this to me suggests, how do we make sense of this, is that all the marker genes expressed in the dorsal CA2 region, which suggests that this neuron SLC1787 RGS14 is present in the dorsal CA2. But since there's no TM2, CAG-Phi, and the weak expression ventral CA2, it means that this cell type has to be absent from the ventral CA2, right? And so one of the ways, the other 7 cell types could exist as kind of a continuum across these axes, but it suggests that the ventral CA2 may just lack a specific cell type compared to the dorsal CA2. So kind of summing up, the gene expression patterns are really useful for identifying anatomical boundaries and cell types, and kind of I was inspired a lot by the CA2 field and seeing the impact that these have made on the research to kind of carry this forward to the rest of the hippocampus. And the thing that keeps coming up to me is this idea of the tissue level patterning versus the cellular level heterogeneity. We can see similarities and differences here, and a lot of them are both important to recognize that they both exist. The new CA2 kind of extends its boundaries way beyond the original, and this idea that there's multiple cell type populations across this axis, I think, is very important. And ultimately, at least maybe 2 subregions defined by essentially the compositions of those constituent cell types and how they're organized. So I'd wrap it up. I want to thank my lab, who we started during the pandemic, and they've kept me sane as we got the lab going. So I want to thank them, our funding, and again, thank the organizers again.